Hi, Miss Nikki here again. We're going to talk about ear and equilibrium. I guess I should say hearing as well. First slide here, just talking about the three parts of the ear. We have an external ear, we have a middle ear, we have an inner ear. The external ear is really just involved with hearing. It's going to funnel those sound waves inward. The middle ear is also going to be involved with hearing. It says tympanic cavity here. We'll talk about what that means in just a second. It's where your tympanic membrane is located, or your eardrum. The internal inner ear, this is where we're going to actually have functions of hearing and equilibrium both. So here's a picture of the regions of your ear from your book. I'm not going to write on this because it pretty much tells you what you need to know. External ear, middle ear, and the internal or the labyrinth of the inside. And we're going to look at where this is located because it's kind of interesting. The first part here, the auricle, the pinna, this is the helix and the lobule. I swear this pen drives me bonkers. Okay, so that's your auricle. Obviously, this is your lobe, and this is your helix. This is called your auditory canal. Some people will call it the external acoustic medius. Most people say auditory canal. And then right here is your tympanic membrane. This serves as the boundary between your external and your middle ear. This is your eardrum, but we're going to call it tympanic membrane, right? This is actually going to vibrate and allow you to actually hear, or allow your brain to recognize that you're hearing something. And we'll talk about this pathway in just a second. Okay, middle ear, words and then pictures. So small, air-filled, mucus-lined cavity in your temporal bone. There are three small bones, the malleus, the incus, and the stapes. We talked about these briefly when we talked about bones. There are three of your inner ear bones, or middle ear bones as we called them. We also have something called an auditory tube. We just talked about auditory canal. They're completely different, so make sure you understand. I'll show you a picture in just a second. Pharyngeotympanic, and that has to do with the pharynx area and the tympanic membrane. That word cracks me up because I don't know anybody that says it. Most people say auditory tube. And this is going to help equalize pressure, and it's better if I show you the picture here. So here is our auditory tube. So right here is auditory tube, and that's this section right here. Here's that tympanic membrane. This is showing you all of the bones of the ear. And then out here, they don't have it on this picture, would be the auditory canal, right? So you can imagine a tricky little multiple choice question about auditory tube versus auditory canal. So make sure you understand the difference. And what happens is, if you've ever been on an airplane and you're trying to equalize pressure, you usually hold your nose and swallow. So if this pressure is high and this pressure is low, it muffles your hearing. You can't hear as well because the pressure difference is causing problems with this vibrating. So usually you swallow, this auditory tube opens and closes again, and when it does that, it allows for the pressure to be equalized from the external and the middle ear environment. Alrighty, inner ear. I love this picture. It's from a different book, but I wanted you to see exactly where the ear is located. So this is a view looking down. So they took off the top of the skull, the clavaria, they've cut it off, and you're looking down into an inferior view of the skull. So you can see the ear here, you can see the auditory tube, and then this apparatus we'll talk about in just a second is what allows you to hear and actually sense equilibrium differences. So it is a bony channel in the inner ear. It has three parts, and they're listed here: vestibule, semicircular canals, and the cochlea. These two are going to have to do with equilibrium, and this is solely the organ of hearing. There's also some fluid in these bony labyrinth areas. They have membrane linings, and they have something called perilymph or endolymph in them. And this will make sense as we move on. Right now, just know there's fluid in these areas. And here's a better picture. So this is one vestibule, and this is another vestibule, and these are the semicircular ducts. So all of this has to do with equilibrium. I'm just going to abbreviate. This circular organ is called the cochlea, and this is involved in hearing. Let's see, what else did I want to say? Oh, the fluid is in here. There's that perilymph and endolymph fluid in the cochlea. Also, if you remember when we talked about cranial nerves, you can see here the facial nerve, the vestibular nerve, the cochlear nerve. Hopefully you remember that the vestibular nerve and the cochlear nerve form the vestibular cochlear nerve, and that goes to the brain. So what are we probably going to be doing here? We're going to have to create some sort of action potential to travel along this nerve to be able to get to the brain in order for your brain to register that you've heard something. So back to the three regions of the ear. Here is, I'm just going to walk through it again one more time. So here is your external ear. What's going to happen? You're going to have sound waves traveling, sounds or sound waves traveling. They're going to hit this tympanic membrane. The tympanic membrane is going to vibrate. That's going to cause these bones to move. That's going to actually cause the fluid that's in the cochlea to move, and then we're going to see what happens next. So before we get on to what hearing is, we need to know what sound is. We need to have some basic definitions. So sound is any sort of pressure disturbance. It's alternating patterns between high and low pressure, and they're going to vibrate an object. And there's a good picture coming up with a tuning fork in just a second. So a sound wave moves outward in all directions, and it's usually illustrated by what they call an S-curve shape. We're not going to get into that too much. I'll basically mention it. So here's a tuning fork, and this tuning fork has been hit by an object right here. What happened was there were all kinds of areas of high pressure or low pressure, but before they became high or low, they were just normal. There were just molecules, these little molecules in the air hanging out. What happens is when this object hit the tuning fork, it pushes all of the molecules into a confined area. That's what these are. So these would be areas of high pressure. Well, if you took everything that was in this area and squished it to one side, what's going to happen in this area? It's going to be low pressure. You're not going to have as many molecules of air in that area. You're going to have tons in one area and not that many in another. So this is a low, and this is a high pressure area of molecules. So if you looked at it in wave form, that S wave they're talking about, you get this wavelength. So this is the wavelength that's traveling through your eardrum, traveling through your auditory canal, vibrating your eardrum, and causing the fluid to vibrate as well. If you want to know the difference between pitch and loudness, it just has to do with how fast the waves are moving or how tall the waves are. So you can see, let me go back. You can see here in pitch, here's 1, 2, 3. Well, guess what? The red one has 1, 2, 3, 4, 5, 6. So pitch is how many waves within a given period of time. And what do the waves correspond to? High pressure. The top part, high pressure, the bottom part, low pressure. What's loudness? How high or what the amplitude of the wave is. Come on. There we go. Okay, so this is actually talking about the transmission of sound. So we can see those little black wavy lines, right? So these are molecules that have been compressed, either high or low. They're coming in. They make the tympanic membrane move. They cause the inner ear ossicles to move, or middle ear ossicles to move. That causes a wave that travels through the endolymph or perilymph. So this picture is showing you the cochlea right here, and it's saying this is actually the organ of hearing. It is a spiral, conical, bony chamber, just like what's listed on the slide. It's divided into 3 chambers. Again, some teachers will make you learn the 3 chambers, some won't, so I would check and make sure. And it holds or houses something called the spiral organ. They used to say spiral organ of corti. Some people leave off the corti part now. It's just the spiral organ of the cochlea. I'll show you these 3 chambers in just a sec. Okay, here we go. So we have the scala vestibuli, and then we have the tympani, let me get it right, and then we have the scala media. So these are 3 compartments within the cochlea. So this is just a sectioning of the entire cochlea. Then we have this organ called the spiral organ, and it is this area in here. And again, you can see some cells, and you can see a nerve, and you can see a ganglion, which is just a bundle of somas. Where do you think this is going to go? To the brain, right? It's going to travel on the vestibulocochlear or cochlear nerve. So we're going to blow this up again and see exactly how this spiral organ helps us recognize sound. So here is an enlargement. This is something called the basement membrane because it's on the bottom. There's this tectorial membrane which sits on the top, so it's like the ceiling. This entire structure is called the spiral organ. And remember what's happening down here. This is one of those chambers, right? So as the sound waves come in, they're vibrating this fluid. So this fluid is vibrating here. There's fluid over here vibrating. There's fluid over here vibrating as the sound wave moves through the cochlea. What you can see is here's the hair cell, and here are tiny little hairs that extend, and it's connected to a neuron. So these are supporting cells, which again are going to help or assist these hair cells in doing their job. But basically what's going to happen is if this is the bottom of the membrane, underneath my arm, bottom of the membrane, and this is the cell with the little hairs on top of it, right? And we have a roof on the top here. What happens as the fluid on the bottom is vibrating this cell, it's pushing it up. When the hairs push up and bend into the tectorial plate that's on the top, this bending of the hair cells causes an action potential to be created. So basically what's going to happen is these tiny little cells are going to get, hair cells are going to get bent over because they're being pushed up into the tectorial membrane. When that happens, an action potential is going to be created, and that's going to travel on the nerve to the brain. So basically this is the wording of that. Vibrations from the ossicles go through the cochlear duct. The vibrating fluid fills the chambers, causes the basilar membrane, which the spiral organ is sitting on, to vibrate. The cochlear hair cells, called stereocilia, are stimulated by the vibrating fluid in the chambers underneath, and above the hairs is a tectorial membrane. Again, another picture. What's going to happen? This fluid is going to be moving. When this fluid moves, it's going to push these cells up into the tectorial membrane. When the cells bend, it's going to create an action potential that's going to travel on the neuron. The vibrations cause the movement of the basilar membrane. The stereocilia are pushed up into the tectorial membrane. The stereocilia bend, which triggers ion channels to open. This is going to cause a graded potential or a local potential. You're going to release a neurotransmitter. It's going to cause an action potential to be created, and your nerves are going to carry that impulse to the brain. So again, this is the picture from your book. I would make sure that you understand these kind of step-by-step processes. Hopefully by now, I've said it at least 2 or 3 times. Hopefully we can do it, right? So sound travels through the auditory canal. It vibrates the tympanic membrane, which causes the ossicles to vibrate, which then causes the fluid in the cochlea to vibrate. What's in this long tube-like structure? That's the spiral organ. And that spiral organ is going to have those supporting cells, and it's also going to have the hair cells with the little stereocilia. When the bottom of the membrane is moving because of the sound waves that are coming in and moving the fluid, so the fluid's moving, the cell's moving, when it crunches with the top of the ceiling or the tectorial membrane, the hairs are going to bend, it's going to produce an action potential, that's going to be sent to the brain. I know it sounds easy when I say it, right? All right, this very last slide here, I'm going to talk about equilibrium in the next video. I cut this video in half because I just thought it was too long. So I'm going to show you this last picture, and then you'll have to pick up on the next video to hear all about equilibrium. So this is showing you an auditory pathway of the brain, just like last time when I wanted to show you where this information is being sent in the brain. I thought I would share this with you again. So when these signals come in, get the pen working, when these signals, those action potentials come into the brain, again, they're going to synapse with other cells. They're going to travel through your medulla oblongata. They're going to travel through your midbrain. They're going to travel to your thalamus and eventually reach the cerebral, let me spell, cortex. And again, this is going to have something to do with some of your reflexes. Remember, traveling through the thalamus, we said reflexes, there's a reflex center there. If you are hearing something too loudly, you can actually dampen by contracting muscles. You can dampen the amount of sound that comes in and out of your ear. So I just wanted to show you the location where these all end up, which is the primary auditory cortex of the brain. And I hope you enjoyed this lecture, and I'll see you in just a few minutes when we talk about equilibrium. Thanks. Bye.